Okay, this will be a discussion on the topic blood. Now, the first thing to realize about blood is that we describe it as a transport medium. In other words, a way to transport substances through the human system. And the pathway is through blood vessels, another topic for another day. But the point is that this transport medium is responsible for carrying oxygen and nutrients to cells and taking wastes and carbon dioxide away from cells. So this blood is considered a tissue. It's a fluid tissue, so it's technically a type of connective tissue, which was all about a matrix, an extracellular matrix, and cells. In this case, it's very specialized because the cells are going to be blood cells with specific responsibilities. When we analyze blood, we can do it one way by putting blood in a test tube and then essentially agitating it. That's called a centrifuge. And we shake the blood, and we'll notice that when we shake the blood, the solid elements, or the so-called formed elements, will settle at the bottom of the tube, and there'll be this cloudy, whitish cloudy substance that's above the formed elements, and that's called plasma. And the plasma represents a little more than half of the total blood by volume. And the formed elements, which are cells, is a little less than half. Okay, and again, as always, these numbers do not require to be memorized in our class. If you remember that plasma is a little more than half and the formed elements are a little less than half, that's all you're going to need to know for any question that I'm going to ask you. When we look at the plasma is mostly water, probably somewhere in the vicinity of 90%. Again, not a number that needs to be memorized. And within the plasma, we're going to have water and plus or minus about 100 other, what we would essentially call solutes, things that float around in that water, mostly dissolved in that water. So electrolytes. Which you might think of as salts. So that might include some sodium, some potassium, some chloride ions, and others. But we're also going to notice that in addition to electrolytes floating around, we're going to have some amino acids. Some of these have to be carried, and fatty acids. Glucose, hormones, and other components along those lines. And a very important one, some gases like oxygen and carbon dioxide. And another category that we want to spend a few minutes talking about is a category of plasma proteins. So obviously, with the name plasma in front of these are proteins that are typically found in the plasma, and they're going to serve a number of purposes. So we're going to look at those purposes right now. Let's get this back up so we can use the full screen. Plasma proteins. So plasma proteins fall into a number of categories. The largest group is called albumin. And albumin plays a very important role establishing what we call the osmotic pressure in blood vessels. And what we mean by that is that this osmotic pressure is the solute pressure that stays inside a blood vessel. You can imagine that when blood vessels get down to the cellular level, the capillaries there are leaky and allow the exchange of nutrients and waste, nutrients to the cells, wastes from the cells, and the exchange of oxygen to the cells and carbon dioxide from the cells. So that's an important function of these blood vessels. The capillaries are leaky, and the blood pressure will force everything out of the capillaries except these plasma proteins and what we call whole cells, red blood cells and white blood cells, typically. And we're going to be talking about those in a few more minutes. So the important role for these plasma proteins is to establish this osmotic pressure, because that's what's going to ensure that the water that was forced out initially in the capillary will be drawn back in after the exchange has occurred. And when that water gets drawn back in because of this osmotic pressure, then carbon dioxide has a gradient direction to follow, and wastes that have accumulated in the blood or in the cells will be drawn back in along with this water. That's typically described as bulk flow. It sets a direction of movement. So other examples of plasma proteins. So we have our albumin group, and then we have a group that are called globulins. Globulins can be described as alpha or beta or gamma. And alpha and beta proteins, they're often carrier proteins. They have many roles, but carrier proteins in the blood. The gamma globulins are known as antibodies. So we know a little bit about antibodies because, you know, for the last 20 months, all we've heard about is whether different treatments will generate an immune response. And part of that immune response is the formation of antibodies, in this case, specifically for COVID-19 virus. We'll talk more about that when we talk about the immune system. But this is where they would be found. These are described as plasma proteins. So they're going to come in contact with viruses or bacteria or foreign substances in the plasma and be activated against those things in that location, the way that antibodies do. Some other examples of plasma proteins include fibrinogen. And fibrinogen is normally inactive unless signaled by chemicals to initiate, or I don't like initiate, we won't say initiate, to contribute to blood clotting. And we're going to talk later today about what that contribution is. But in between times, if there's no need for blood clotting, this fibrinogen is just part of the plasma protein group. So these are some examples of plasma proteins. Under most conditions, they stay within the blood vessels so they can form this osmotic pressure or this osmotic pull that favors returning water and waste products back into the blood vessels during capillary exchange. Important role to play in that respect. The formed elements and remember, they represented this smaller percentage are cells. And the cells are either red blood cells, about 99% of all of the formed elements are going to be red blood cells. White blood cells, so they're a little bit less than 1%, and platelets fall into this category as well. So we got three different cell types to be aware of. The largest component, clearly, are the red blood cells. So we're going to spend a fair bit of time talking about those red blood cells because they're very specialized cells with very specific responsibilities. So the scientific name for a red blood cell is an erythrocyte. We can abbreviate a red blood cell in our classroom as an RBC. Okay, so that's an abbreviation for red blood cells. And as long as you remember that another name for red blood cells is an erythrocyte, everything is good. Everything will work out just fine as far as that kind of thing goes. So these are very specialized cells. Now, most of us should remember that the process of cell development is called hemopoiesis. We talked about hemopoiesis when we talked about bone tissue. So in this picture here, we can see that we have a stem cell. The stem cell is simply another way of describing an undifferentiated blood cell. This is sometimes called a hemopoietic stem cell. It hasn't yet differentiated. When it begins to differentiate during embryonic development, we're going to see it go in two directions. We see a myeloid lineage, and we're going to see a lymphoid lineage. The myeloid lineage is the one responsible for most of the cells we typically think about when we think about cells in blood. So that's going to include our red blood cells. It's going to include a group of white blood cells that are categorized as granulocytes. And we can also add in, we're missing one in here. We're going to throw another one in here called monocytes. So these all fall under the category of a myeloid stem cell origin, or sometimes called a lineage. Lineage just means that that's the pathway, the line of development that it followed. So red blood cells are going to be the first group that we talk about. When we look at the granulocytes, they're called granulocytes because they have granules that show up. You can see in this eosinophil, there are red granules. In the basophil, they're described here as purple, and they're kind of pinkish in the neutrophil. Because they have granules that show up under a microscope, when they're stained properly, they're described as granulocytes. The lymphoid cells are agranular. They don't have granules in their cytoplasm, and so they're described as being agranulocytes. They are absent of granules. That's what the A in front of it means. And the monocytes fall into that category as well. When we look at those under a microscope, they don't have any granules to show up for. And the other category to be aware of are our platelets. So our typical white blood cells, our platelets and our red blood cells come from the myeloid lineage, and white blood cells that are active in the immune response because they can generate antibodies in the case of B cells, and they can directly attack cells in the immune system to kill a virus directly or a bacteria directly or a cancer cell. Those are called T lymphocytes, and they come from the lymphoid lineage. So they all originate from a hemopoietic stem cell, but then separate to follow a separate pathway. Okay, we'll talk a lot more about these lymphoid stem cells and the cells that come from them when we talk about the immune system. So we want to spend a little bit of time looking at red blood cells. So their primary responsibility is to carry oxygen in the blood to all cells. Now, specifically, all cells that require oxygen in order to make lots of ATP. So the process of a cell making ATP is called respiration, cellular respiration. And that refers to the fact that oxygen is necessary in order to make lots of ATP. So we'll come back to that later on as well. So in order for this cell to be good at delivering lots of oxygen, it becomes very specialized. So a mature red blood cell is described as having no nucleus. If it has no nucleus, it cannot undergo mitosis. And if it cannot undergo mitosis, it has a defined lifespan. In other words, eventually it's going to die. So that defined lifespan is anywhere from about 90 to 120 days, 3 to 4 months. And therefore needs to be replaced. So the red blood cell lineage that we talked about, that we all know goes on in bone marrow, is an ongoing process. This is happening all the time. There are trillions of red blood cells circulating in the body. And at any given moment, some are dying and others are replacing them. So it's a responsibility of a hormone that we're going to talk about in just a few more minutes that will ensure that the bone marrow is being signaled to continue to make enough red blood cells to meet demand. So this red blood cell is very unique because it's really the only example that I can think of right at this moment of a cell that doesn't have a nucleus. That's a very rare scenario as far as that goes. So what else do we know about these red blood cells? The term is anucleate. And again, that A in front of a word just means an absence. So that just means no nucleus. So we know about that already. That's one of the features that's unique. It has no mitochondria. So that seems a bit unusual. This is a cell that delivers oxygen to all other cells so that oxygen and glucose together can make lots of ATP, but the red blood cell itself has no mitochondria, which means it has no ability to generate lots of ATP. So by definition, it can only make ATP anaerobically, which simply means without oxygen. So that means very little ATP is going to be produced. But a red blood cell, once it's mature, doesn't need to make very much ATP because its role is to carry oxygen. It doesn't need to move. It doesn't need to undergo mitosis. It doesn't need to go through endo or exocytosis, any of those things that might require ATP. And a mature red blood cell, let's make sure we realize this is mature. Red blood cell has no other organelles. Once a red blood cell is mature and has no nucleus, there's no direction for protein synthesis or anything along those lines. So we don't need a rough endoplasmic reticulum or any other the types of organelles that we would find. Instead, the cytoplasm is filled with a protein-based molecule called hemoglobin. This is an oxygen-carrying molecule. In one red blood cell, there will be somewhere in the vicinity of 250,000 hemoglobin molecules. Each hemoglobin can bind, which means carry, 4 oxygen molecules. So for those of you who are mathematically inclined, you will realize that one red blood cell carries about 1 billion oxygen molecules. And I mentioned earlier that there are trillions of red blood cells that circulate in our blood. One cell is microscopic, and it's essentially gotten rid of everything in its cytoplasm except hemoglobin in order to be very, very good at carrying oxygen. Now, again, the numbers don't matter. The concept, though, is that there is a lot of oxygen being carried on these hemoglobin molecules within a red blood cell, and there's a lot of red blood cells. It tells you how important delivery of oxygen is to all the cells of the body. It's the most numerous cell in the blood. It has huge capacity to carry oxygen, and that tells you that it must be very, very important to carry oxygen. And these red blood cells determine what's called the hematocrit. The more red blood cells in blood, the thicker your blood gets. And that can be a problem. If blood gets too thick, it becomes difficult to travel through smaller blood vessels. So the numbers of red blood cells become regulated, closely regulated, like everything else does in homeostasis. And the hormone that's responsible for this regulation is called erythro. So you see erythro, you're thinking red blood cell. And then the hormone is called erythropoietin. It's got an abbreviation, EPO. It's produced by cells in the kidney and stimulates the myeloid cells in bone marrow to produce more red blood cells. So the kidney is responsible for filtering blood. If the kidney determines through filtering of blood that the numbers of red blood cells are going down, or more importantly, not enough oxygen is being delivered through the body, then it's going to release this hormone, erythropoietin, which will target the bone marrow and encourage more production of red blood cells. Okay, so anytime oxygen delivery is reduced, EPO is going to get released to stimulate more red blood cell production. And when EPO stimulates increased red blood cell production, we see increased numbers of a cell called a reticulocyte. And a reticulocyte is an immature red blood cell. This immature red blood cell will have a nucleus, and it will have some mitochondria and some rough endoplasmic reticulum. This is the cell that can start to produce hemoglobin because it's got rough endoplasmic reticulum and a nucleus, and it can undergo a little bit of mitosis as well. But eventually, as the hemoglobin concentration increases, the nucleus is going to disintegrate, and the mitochondria will disintegrate, and the rough endoplasmic will disintegrate, and what we'll be left with is a mature red blood cell that is empty of all the organelles and filled with hemoglobin, so that it now becomes a very effective cell for carrying and delivering oxygen to all the other cells of the body. Now, the other cell types that we have in the body are white blood cells, and their scientific name is leukocytes, which are white blood cells, WBC. And we can put a little s on there to make it plural. So these are much lower in numbers. The red blood cells are about anywhere from around 99% of the formed elements. I think I mentioned that earlier. It's a ratio of about 1,000 to 1, red blood cells to white blood cells. But these are very important cells for defense. These cells are part of the immune system. We'll talk more about them when we talk about the immune system. But for now, what we want to realize about our leukocytes is that these are technically the only complete cells circulating in the blood. Complete because they have a nucleus, they have endoplasmic reticulum, et cetera, et cetera, et cetera. Mitochondria, they can undergo mitosis, so their numbers can change quite dramatically. If we're fighting an infection or fighting an injury, these white blood cell numbers will change. They will go up in order to recruit more cells in defense of whatever it happens to be. So these cells are mobile, meaning they can move in and out of blood vessels under the right conditions. And they're protective. Mobile protective cells. So red blood cells always function within the bloodstream and don't leave the bloodstream unless there's an injury, whereas white blood cells can move in and out of blood vessels depending on whether they're needed in tissues for infection purposes or repair purposes, things along those nature. So whenever these white blood cells are required for defense, their production increases. White blood cells can double in number within a few hours. So if there's the presence of a virus or an injury to the tissue, white blood cell numbers are going to increase dramatically. There are chemicals that will be released by the damaged tissues that will signal an increase in mitosis of these cells. So we mentioned when we looked at the diagram that white blood cells, let's move this up. We want a bit of space here to talk about our white blood cells. We can talk about our white blood cells in terms of the cells that were called granulocytes. And they have names like neutrophils, eosinophils, basophils. We can even include some mast cells here. They're all called granulocytes because if we look at them under a microscope, they tend to have a nucleus that's often got lobes associated with it. But more importantly, when it's called a granulocyte, we can see granules in the cytoplasm. Now, in some cases, these granules are enzymes that when they're released will contribute to an immune response, a defense response. And sometimes they're chemicals that will promote inflammation or promote platelet activity, that sort of thing. So granulocytes show up with granules. And these are all white blood cells that will be involved with defense. And they'll have processes like phagocytosis, or they will promote inflammation, or they may promote blood clotting. All things that contribute to the defense of our system. Agranulocytes include our lymphocytes and cells that have a great name we'll talk about when we do the immune system, natural killer cells, and those monocytes. So when we look at an agranulocyte under a microscope, we've got a nucleus, and the cytoplasm appears clear. So that's really where the category differential is. These have a variety of immune responses. That we'll talk about when we talk about the immune system. Monocytes are literally mega drew a little mental block there, not mega, but they're called a macrophage, which is literally saying they're a big eater, macrophage. So under the right circumstances, when they move out of the blood and into a damaged tissue, they become very voracious eaters. They're going to clean up the environment. They're going to eat virus or dead tissue, clean it all up. So these are the categories of white blood cells that we see circulating in the blood that can increase in number very dramatically when they're called upon for action in defense roles. The third category of formed elements are platelets. And they're derived from a cell that's called a megakaryocyte. And that was part of the myeloid group. But they are fragments of this megakaryocyte. So they circulate around in to the tune of maybe a few 100,000 in a milliliter of blood. So very small contribution by volume, but they can increase in numbers anytime where we need blood clotting to occur. These platelets are have no function per se unless there's an injury to a blood vessel and we need blood clotting. Then they become activated. So they circulate in the blood, minding their own business, and then in the case of blood clotting, they become sticky and increase in numbers, and they can help block the site of blood loss and contribute to the repair process of the damaged blood vessel. We're going to be talking about that in just a few minutes. Okay, so when we look at the formed elements in blood, we're talking about red blood cells, white blood cells, and platelets. Okay, and the formation of these blood cells follow two distinct lines, starting with a myeloid cell that's responsible for red blood cells, the granular white blood cells, neutrophils, basophils, and eosinophils, and monocytes and platelets, all from the myeloid lineage. The lymphoid lineage is responsible for T cells, T lymphocytes, and B lymphocytes, natural killer cells. They're the big ones that are going to come out of the lymphoid lineage. And those are the ones we'll spend some time talking about when we talk about the immune system. Okay, now I mentioned earlier that we would talk about blood clotting and the role that different cells play in the process of blood clotting. So the fancy word for blood clotting is called hemostasis. Hemo, so you know that's something to do with blood when you see that hemo. Stasis, which literally means stopping blood. So hemostasis is the stopping of blood, in particular blood loss. What we're concerned about with blood loss is the inability to deliver oxygen to all the cells at the level that they require it. So we can survive a little bit of blood loss for a short period of time, but we can't survive an extended period of time with blood loss. When we start to talk about losing blood volume to the tune of 15, 20, 25%, we're going to start to become weak and pale. And if we start to lose anywhere above 30%, it becomes life-threatening unless we can stop it. So normally, blood flows within blood vessels in what is called a closed circulation, which really means no exposure to outside stimuli. But if it's damaged, there's a necessary reaction, series of steps that has to occur to stop that blood loss before it becomes life-threatening. And that's the process that's called hemostasis. It involves many substances that are found normally circulating in the plasma. We know some of those. Platelets are one of those things. Fibrinogen is another one, circulating in the plasma. Okay, but they, as we also know, they're inactive unless they're stimulated. So there are 3 stages or steps to hemostasis. The one begins with platelets, and we call that a platelet plug formation. So literally, like it sounds like, the platelets are going to get clumped together and they're going to plug the area of damage. Then we're going to see almost at the same time, and some textbooks will tell you this one happens first. It doesn't really matter. Vascular spasm. That means the blood vessel is going to constrict, very aggressively constrict, with the goal of trying to minimize blood loss, right? If the blood vessel's dilated, more blood's going to flow. We're going to lose more blood. So if we say a blood vessel normally looks like this, and if we cause injury and break that blood vessel so that blood is now able to escape, we're in danger of blood loss. 2 things are going to happen. We're going to plug it up with platelets. We're going to talk about this in a second. And we're also going to vasoconstrict. We're going to constrict the blood vessel to try to minimize blood loss and allow the plug to become stronger. We said there were 3 phases. The 3rd phase is the process of coagulation. This is a series of steps. 1 step before the 2nd step, before the 3rd step can occur. And this is an example. Here's an example of positive feedback. Coagulation and the steps required for coagulation are an example of positive feedback. This is not homeostasis. This is a situation where, as long as there's blood loss in that blood vessel, the steps for coagulation will get stronger and stronger and stronger. Once coagulation results in a complete plug, then the positive feedback stops. So it's temporary, and that's part of a definition for positive feedback when it happens in the body under normal conditions, like birth. Once the baby is born, the positive feedback that occurs in the uterus stops. In coagulation, it stops once the blood vessel is repaired, or at least plugged before it's completed its repair in a lot of cases. So when we look at the steps, Okay, where are we here? If we look at platelet plug formation, we should all realize that the platelets are going to be part of this process. And what's going to happen during platelet plug formation is the platelets are going to become sticky. So normally, normal platelets are small and repelled away from blood vessel walls. Which is called an endothelium. It's a type of epithelium. It's inside endothelium. So normally platelets are repelled from the endothelium. But if the endothelium gets broken, when the endothelium is damaged, chemicals are released that signal platelets to become active. Platelet activation. And when these platelets are signaled to become active, they are going to become sticky and start to clump together. And they're going to be drawn towards the site of injury because where that interruption occurred, there'll be increased flow towards that area. The platelets will be drawn to that area. They're sticky and they accumulate and they form a plug. And this happens within seconds. So it's a very fast process, which of course is important because we're trying to minimize blood loss. Now I'm having trouble getting rid of this for some reason. Hemostasis. That would be why we got onto the wrong layer. Okay. So let's get back to where we were. We were talking about platelet plug formation. Now we want to talk about the next two phases. We're going to show you that picture in a minute, so we'll come back to that picture. So the second two phases, the other thing we want to talk about, so we had our platelet plug. That happens within seconds. And almost simultaneously, we get our vascular So again, typically signaled by the chemicals that get released from the damaged tissues. Collagen is exposed because the underlying connective tissue has been damaged, and collagen is exposed, and it releases signals and activates chemicals in the blood to release chemicals to signal both platelet plug formation and vascular spasm, especially a chemical called serotonin. That's released from platelets to cause blood vessel spasm. Okay, so that B dot V dot is blood vessel spasm. So signaled by this serotonin. And that's going to decrease the blood loss until the clotting, the coagulation can actually occur. This is going to occur because the smooth muscle cells in the wall of the blood vessel, stimulated by the presence of serotonin and other pain receptors, will spasm and close off the blood vessel. Not completely, but reduce the flow through that area. And that sets us up for coagulation. And coagulation is a fancy word for clotting. So in coagulation, there's going to be these series of steps. I mentioned earlier that it's an example of a positive feedback mechanism. So there are chemicals that get released. So we start, for example, with the injured blood vessel tissues, release chemicals to initiate the steps of coagulation. So we have factors that are released from the damaged tissues, and we have platelet factors, factors that are released from within the blood vessel itself. So we're going to end up seeing two pathways. One pathway is described as the intrinsic pathway because it starts within the blood vessel. And the other pathway is described as being extrinsic because it starts from signals that came from the damaged tissues outside the vessel. So what we're looking for when we look at these steps is to understand a couple of things. So the first thing is injury to the blood vessel lining. We don't like that, where that went there. Lining, which we call the endothelium. And that exposed collagen and underlying smooth muscle cells and so on, and that activated the platelets. So we have a series of platelet factors. And they're often described as a cascade. So there's a series of steps. Some of you maybe have heard that, this is where some factors like a factor 3 and a factor 8 and a factor 5. We're not going to worry about the factors. We're going to understand that we have an intrinsic pathway happening within the blood vessel, and we have an extrinsic pathway that happens from outside of the blood vessel and has a series of steps that are necessary. So these would be tissue factors. So I'm purposely not giving you the factors, okay? But what we recognize here is that these steps require calcium. That's important. They require calcium. And the goal of both pathways, intrinsic and extrinsic pathway, is to form what's called prothrombin activator. We'll just throw this one in as well, vitamin K. So the two big requirements in order for blood clotting to be successful is enough calcium and vitamin K. And the goal of the two pathways is to ultimately form prothrombin activator. In other words, we can form prothrombin activator from two directions, but when both directions are working, it's better. Okay, and then what's going to happen is prothrombin activator, this shouldn't be a big surprise, is going to act on something called prothrombin. And the prefix pro tells us it's like pre, it's not yet active. But when prothrombin activator acts on prothrombin, it becomes thrombin. And when thrombin forms, thrombin acts on fibrinogen. We mentioned fibrinogen earlier today to become fibrin. And fibrin is an insoluble protein that will be able to form a mesh around all those platelets that created the early plug. And that mesh now can trap red blood cells and fibroblasts and allow a concentration of cells and proteins to encourage repair of the damaged blood vessel. So we trap red blood cells and we trap fibroblasts. The fibroblasts can create new collagen, fibrin, and new collagen, I should say, and elastin, and deposit some extracellular matrix. It's going to encourage the repair process. And we're going to get a more complete repair. So this process takes minutes. So the platelet plug that initially formed is seconds. The coagulation steps are minutes. And then the actual remodeling and reorganization and repair process, it could take anywhere from days to months. And we know a little bit about what happens is if this blood vessel or the damage to the skin came out to the surface, we would get the formation of a scab, right? And we know what happens to a scab eventually. It dehydrates, and that's when it starts to feel itchy, and eventually falls off once the skin underneath has repaired itself. So the scab is part of this clot that we've formed. And it dehydrates, which helps pull the damaged edges of the skin closer together and then remodels to repair, and the scab falls off. But we know that can take days and sometimes weeks before it completes itself. But the idea is we've closed off the area where the blood loss was occurring, and we minimize blood loss. So here's what we're looking at when we look at these three steps that are going on. In our picture here, we can see that the tissue has been interrupted. So the collagen has been exposed. The platelets that would normally circulate around minding their own business become activated, so that the idea is we're going to minimize the loss of red blood cells by clumping. Vasospasm and clumping of the platelets blocks the blood loss. Then we get the steps for coagulation. Damaged cells, extrinsic, platelets, intrinsic, the formation of a prothrombin activator, which converts prothrombin to thrombin, fibrinogen gets converted to fibrin, and we form a nice, strong clot that can trap red blood cells and fibrocytes to be able to initiate and complete the repair process. All of that's going on in those three steps: platelet plug formation, vasospasm, or blood vessel constriction, spasm, and then the coagulation process, the steps that occur in order to encourage a complete plug. I don't want you to spend time on these diagrams here, but recognize in this picture that what we're looking at is an intrinsic pathway and an extrinsic pathway. Tissue injury within the blood vessels themselves, changes within the blood vessels, like the exposed collagen that starts the pathway. Down we go, down we go. We need calcium. Prothrombin activator is the goal of both pathways. In order to produce prothrombin, we need vitamin K. So that's going to be an important factor in the production of prothrombin that gets converted to thrombin, fibrinogen to fibrin, and we get the formation of an insoluble clot. Okay? Everything you see in these blue boxes here are medications that have different effects on this process. Nothing that we need to worry about in this class. Okay? This is just to illustrate the two separate pathways that get us to the prothrombin activator. In other words, the goal of the intrinsic pathway and the goal of the extrinsic pathway is to get us to prothrombin activator. All right, that's the steps that we need to know a little bit about for hemostasis. Finally, for today, we're going to talk about blood groups. Most of us, especially because we're interested in healthcare, are generally aware that bloods have a type. We talk about blood groups or blood typing. And basically why this is important is that if or when blood loss occurs, we know that the bone marrow will increase. So bone marrow cells which we call them myeloid cells, will produce more red blood cells. So that's good. If it's a mild blood loss, not a big problem. But like I said, if we have somewhere between 50 and up to say 30% blood loss, we're going to get weak and pale because of reduced oxygen delivery. More than 30% blood loss could cause severe shock, which could lead to death. So this whole blood clotting thing is important because in lots of cases, we may not produce enough red blood cells fast enough to make up for this loss. So sometimes a whole blood transfusion and if it's a whole blood transfusion, it's replacing all the cell types. It will include red blood cells, white blood cells, and platelets that can be, you can sometimes just do a red blood cell transfusion, um, uh, which is a little bit more specific. But if we're going to provide a patient with a blood transfusion, we have to be aware of the patient's blood typing versus the donor blood typing to prevent rejection. If we mix the bloods and we have a mismatch, the blood will clump, the blood will react, our immune system will react. We have antibodies for the other blood types. And what's really unique about blood groups is that red blood cells are identifiable by one of really three types. They're identifiable by the letter A, the letter B, or neither of those two. So the presence of an A or a B or none actually will allow us to have four types. We can have type A, we can have type B, we can have neither, which we traditionally call type O, which more correctly would be type 0, but we call it type O. And then the fourth type is a combination of A and B. So the key thing to recognize here is that when we look at a red blood cell, so a red blood cell is an oval shape. We know that it doesn't have a nucleus. We know that it's full of hemoglobin. And the hemoglobin is binding oxygen so that it can carry through the system. So for example, if we talked about type A, what we're essentially saying is that there are markers on this cell membrane that identify it as a type A, and a type B would look like this, a cell that has the B marker on it. So these are identifying markers on these cells. The type A cell is different than the type B cell. And then we have another cell type that has no markers on it, and a fourth cell type that has both on it. So this is, so far, what we talked about is pretty typical. So this would be our type O, and this is our type AB. Okay, so every cell in our body has cell markers on it that identify them as self cells. In the case of blood, there are, and a typical cell will have hundreds to thousands of markers on it, which is why tissue transplant is so complicated. Tissues have hundreds of different markers that need to be matched in order for a donor to be accepted. In the blood typing, there's really only two main markers, an A and a B, that will have an aggressive reaction if they're mismatched. So the idea here is that if we take a look at our red blood cells, okay, if we give ourselves a chart. So here's our chart. And if we said that our red blood cell marker, which is also described as an antigen, so AG, full word is antigen. So what we're saying is that if the person is type A blood, they've got an A marker. Somebody with type B blood has a B marker, no antigen. And we could have somebody that has both. Okay, so our red blood cell is identified this way. Here's the trick. Here's what's very unique about blood typing. The plasma is our location for antibodies. Now, normally for antibodies to form, we have to have exposure to the antigen. So in other words, if we've been exposed to the virus that causes COVID-19, we're going to develop antibodies for COVID-19, but we didn't get those antibodies until we had an exposure. In blood typing, we develop the antibodies before the first exposure. So we've got antibodies in somebody with type A blood has automatically got antibodies for B. And somebody with type B blood automatically has antibodies for type A. This is very unusual, but it's very significant because we recognize right away then that if we were to give somebody with type A blood B donor blood, we would get a reaction. This represents a mismatch. So in these two situations, we've got a mismatch. We cannot give somebody with type B blood A blood because it's going to be a mismatch. It's going to create coagulation, clumping, could potentially be life-threatening. Now, somebody who has AB blood, they have both antigens, A and B. By definition, they will have no antibodies. Because if they did, they would react against themselves. But the person that has no antigens, or type O blood, has antibodies for both. So when we look at this little chart, we can call the type O blood person is somebody who's referred to as the universal donor. Because if they give blood, there's nothing on the new red blood cells to stimulate an antibody response. There's no antigen to cause the antibodies to react. So that person, a type O person, becomes a universal donor. No antigen for A or B. Okay, but the trick is they can only receive blood from another type O because they have antibodies for both. So universal donor, but not a universal recipient. The universal recipient is somebody who has no antibodies. If you have no antibodies, it doesn't matter what blood type you are given. There's nothing to react to the new blood. So antibodies are going to react to the opposite blood group. But if there's no antibodies, there's no reaction. So this person can receive blood from all of the other three types, type A, type B, or type O, because they have no antibodies, so there will be no reaction. So your blood type is dependent on which antigen your mom and your dad gave you. Okay, and in next semester, you'll talk a little bit about genetic inheritance and things like that. So that could become something you'll understand better in terms of your parents providing you with genetic coding, genetic presentations that determine lots of factors. You know, the color of your eyes, the whether you have a dimple on your chin, you know, whether the index finger is taller than the fourth digit is. You know, those sorts of things are all genetically determined. So is your blood typing genetically determined. A baby who's born with type A blood will form antibodies to type B blood automatically, and therefore cannot receive type B blood from a donor because there would be a reaction. Now, the other blood type that's important for red blood cells is Rh. So the point to make here is typical cells have hundreds of markers to identify self, which makes transplants complicated. That's why when you hear about somebody who's looking for a new kidney or a new lung, they put out a request. Closest relatives can get tested first because they're more likely to have a better match. And then you can go into communities. Like if you're of Greek origin, there's a better chance that Greeks will have a better match, but no guarantees because there's hundreds of these markers that need to be identified. And in typical cells, any one of those hundreds can cause rejection. But in red blood cells, there's really only two. We've already talked about the ABO, and the other is called the RH factor. So if you have the RH factor, and again, here's your blood vessel, you could be a type A. And if you also had the RH factor, this person would now be described as being a type A positive because they're positive for the Rh factor. The person below here that has type A blood, but no marker, is type A negative. So there's only two things to worry about with respect to the Rh factor. Either you have it, positive, or you don't. So the problem can occur if mom is Rh negative, but she carries a baby that's Rh positive. Now, in the first pregnancy, that's not a problem because mom has not yet been exposed to Rh positive, so she doesn't have antibodies yet for Rh positive. So the Rh factor does not automatically generate antibodies like type A does or B. So the Rh factor is more typical. You have to have a first exposure to develop antibodies. So when a mom who's Rh negative carries a baby who's Rh positive for her first pregnancy, everything is normal, but mom now has had an exposure to Rh positive, which means she will now have antibodies to Rh for a second baby. So if she's not treated to inhibit the immune system from forming those antibodies, or if the baby isn't treated in the second pregnancy, there's the possibility that mom's Rh antibodies will attack the baby's Rh positive red blood cells. And when that happens, it's called hemolytic disease of the newborn. We're not going to spend a lot of time on that because that's a pathology. Talk a little bit more about that when you get into PNA 300 and 400. But the idea, the thing to be aware of is first pregnancy is probably safe. Second pregnancy could be a risk. Mom is Rh negative. This means that dad must have been Rh positive to have a baby that's Rh positive. So the baby will be born with anemia, meaning it will not have enough oxygen being delivered to its tissues, and it needs to have transfusions right away in order to survive. Okay, so that's a very specific situation. That was called hemolytic that's due to a mismatch between Rh negative mom and Rh positive baby, especially second or after born. Okay, not so much on the firstborn. Okay, that's the kind of stuff that you need to know about for this semester and blood.